Hello, good evening everyone, and I hope you all have digested the truth that the NEET PG exam got postponed for a while. But still, we should look all these things in a positive perspective. Now tell me, what is the most challenging topic, whether you are going for USMLE, NEET PG, MBBS exam or anywhere? Urea cycle. Oh my God. We don't like all those enzymes to be remembered. We can't remember in the real exam, but there is a secret. In the next 5 to 6 minutes, I'm going to make you the champion on the urea cycle. Shall we start? Wonderful. So, the most important mnemonic that you need to remember about urea cycle is one Coca-Cola bottle, one coffee, of course, alcohol of Argentina for an aggressive urine. You'll remember them pictorially, right? So, what is the mnemonic? Orange cola, coffee, alcohol of Argentina for fumarate, aggressive urine. This is what you need to have in your mind. A bit of coffee, a bit of cool drink, a bit of wine will make you to remember the urea cycle. Orange, please recite along with me. Orange, ornithine. Cola, carbamoyl phosphate. Coffee, citrulline. Alcohol, aspartate, which enters the cycle. Argentina is arginosuccinate. Fur is fumarate that leaves the cycle. Aggressive is arginine, and urine is urea that leaves the cycle. Very simple. Now you are the champions. So, the two nitrogen atoms of the urea, from where are they derived from? So, you should remember, ammonia gives one nitrogen and the other one comes from aspartate, also donates the another nitrogen. And from where does the carbon atom of the urea comes from? It is supplied by this carbon dioxide. That is what you have to remember. Two nitrogens and one carbon. So basically urea synthesis, 5 minutes you need to remember because it has 5-step cyclic process. 5 distinct enzymes. And 2 enzymes are in mitochondria and remaining 3 are in cytosol. So commonly asked question in the exam, what are the enzymes of urea cycle in the mitochondria? What are the enzymes in the urea cycle which are there in the cytosol? You need to be very sure about. Now if you look at the first step, what is the first step? It is the formation of this carbamoyl phosphate. And remember, what is the frequently asked question? It is rate limiting. So carbon dioxide plus ammonia plus 2 ATPs. 2 ATPs are consumed, please remember. You will get all these notes PDF in the Telegram group. So please don't forget to join the Telegram study group. Knowledge is absolutely free. And I keep giving you a lot and lots of short videos, 5 minutes, 6 minutes short videos. And I will send you an update if you are a part of the Telegram group, right? Now, carbon dioxide plus ammonia plus 2 ATPs consumed will give you carbamoyl phosphate, and for this you require N-acetyl glutamate, which is an allostatic activator that stimulates the carbamoyl phosphate synthase 1 is what you need to remember. Now one big important point you should not forget. to make our life more miserable. There is one carbamoyl phosphate synthase 1, carbamoyl phosphate synthase 2, like Jumanji part 1, Jumanji part 2, like that. So what are the favorite questions of the examiner? Where is CPS-1, CPS-2? CPS-1 is in mitochondria. CPS-1 belongs to urea cycle. How will you remember? You will tell the teacher, teacher, little finger, you say. One finger is urine. Urine is urea cycle. That is CPS-1. CPS-2 is in the pyrimidine biosynthesis, and it is there in the cytosol, and it uses the glutamine, whereas urea cycle uses ammonia. And this CPS-1 is activated by NAG, N-acetyl glutamate, whereas CPS-2 is inhibited by the end product, feedback inhibition, by the CTP. That is what you need to remember. Now the second step, come on, quickly tell me. It is the formation of the citrulline. This is also mitochondrial. Ornithine plus carbamoyl phosphate is equal to citrulline. And what is that enzyme called? Ornithine transcarbamylase is what you need to remember. Ornithine and citrulline are basic amino acids. You don't find them in the protein structure because we don't have a codon for that. In fact, the citrulline is transported to the cytosol by a transporter system. Citrulline is not there in tissue proteins, not in the blood, but citrulline is there in the milk. That's what you need to remember. Now, step number 3, formation of the arginosuccinate is what you need to remember. Citrulline condenses with aspartate. Aspartate comes from outside. Aspartate to form arginosuccinate, and that's the reason the enzyme is called arginosuccinate synthetase is what you need to remember. And this is how the second amino acid of the urea, amino group of the urea, got incorporated. But this requires ATP, and it is cleaved to AMP. So 2 high energy bonds are required in this step. That's what you need to remember. Then the arginosuccinate is broken down by arginosuccinase. It is also called arginosuccinate lyase. It breaks down the arginosuccinate to arginine, and once more fumarate is regenerated. And fumarate is the intermediate of citric acid cycle, all of you know very well. So that is the reason fumarate is called the connecting link between the citric acid cycle and the urea cycle, is what you have to basically remember. So what is the fate of this fumarate? Fumarate, by the action of fumarase, become malate, become oxaloacetate, once more regenerating the aspartate which is required for the urea cycle to run. So, but fumarate is good. It is like petrol. You sell petrol, you get money. You sell petrol, you get money. So, fumarate will lead to the formation of the NADH, which is the source of energy generation once more. So, that is the reason you should know how many ATPs are consumed, how many ATPs are produced, and what is the net expenditure for the urea cycle to run. We will take it up next. So, the final step is the formation of the urea. Arginine is broken down by the enzyme arginase, which is the final enzyme that breaks down the arginine and yields the urea and ornithine. And ornithine is regenerated, it enters the mitochondria, and it is reused in the urea cycle. And this arginase fellow, who is leading to the formation of the urea and ornithine, it is activated by the cobalt and manganese, is what you need to remember. Then this ornithine and lysine, They both compete with the arginine. So the end product ornithine and also lysine, these two will have a competition with the arginine for the arginase is what you need to remember. So the end product is a competitive inhibitor, feedback inhibition. So where do you find this arginase? Arginase is found in the liver. Whereas all the remaining 4 enzymes of the urea cycle are present in all other tissues. That is the reason, because only liver has arginase. That's the reason only in the liver when the arginase functions, urea is produced. That's the reason urea cycle enzymes are present everywhere, but arginase is present only in the liver. That is the reason urea ultimately is produced only in the liver, because arginase is there only in the liver. Gotcha? That's great. So finally you should know the beautiful compartmentalization created by the Almighty. The first 2 enzymes are there in the mitochondria. The first 2 enzymes, what are the first 2 enzymes? Carbamoyl phosphate synthase 1, ornithine trans carbamylase, they are there in mitochondria. And fumarate inhibits the 4th step, which happens in the cytosol, but the fumarase is there in mitochondria. And arginosuccinate lyase is located in the cytoplasm. That is what you need to remember. So finally, guys, let us talk about the energy of the urea cycle. So that by remembering a Coca-Cola, by remembering a coffee, by remembering a Argentina wine and the urine, you can remember urea cycle enzymes. So what is the energetics? Ammonia, carbon dioxide, aspartate will lead to urea plus fumarate released. 2 ATPs are used in first reaction. Another ATP is converted to AMP, so 2 ATPs are also gone here. So this 2 ATPs, 4 ATPs. But the fumarate formed in the 4th step is converted into malate. Malate will lead to production of 1 NADH by the action of malate dehydrogenase, and that lead to 2.5 ATPs. So 4 minus 2.5 is equal to 1.5 is the ultimate energy expenditure if you want to run the urea cycle. And always remember, urea cycle, citric acid cycle, they both are connected with one another, and that is called urea bicycle. So let us finally look at the regulation of the urea cycle. Carbamoyl phosphate synthase 1 is the rate limiting enzyme. It is allosterically activated by N-acetyl glutamate. In my next video, I will talk about all the urea cycle enzyme defects and what are the disorders, how do they clinically present, how do you treat, what happens in hyperammonemia, everything in my next video on this YouTube channel. So don't forget to subscribe to the channel, click on the bell sign for the notifications, and also don't forget to join this Telegram study group. You get thousands of friends across the world preparing for the medical licensure exams. So, how is the urea cycle regulated? So basically, N-acetyl glutamate is the controlling substance. Key is there in its hand. And this N-acetyl glutamate is formed from glutamate and acetyl-CoA, and it is being synthesized by a synthase and degraded by a hydrolase. So it is all this N-acetyl glutamate concentration which will decide the rate of the urea synthesis in the liver is what you need to remember. So acetyl-CoA typically become CoA and glutamate become N-acetyl glutamate. This is what you need to remember. So any high concentration of the arginine, increase the N-acetyl glutamate. Any protein-rich meal increase the level of the N-acetyl glutamate. That is the reason any high arginine or high protein diet will lead to increased urea production. And both the carbamoyl phosphate synthase 1 and glutamate dehydrogenase, they both are there in the mitochondria. They closely coordinate with each other with regard to the formation of the ammonia and its utilization for the carbamoyl phosphate production for the urea cycle. So that is all you should remember. Coca-Cola, a coffee cup, and an Argentinian wine, and the statue pissing as a mnemonic to remember the urea cycle. So when you promise me that you will master this cycle and get ready for the tomorrow's exam. Always yours, Dr. Murli Bharadwaj here.